The human musculoskeletal system is rarely taught accurately. It is typically presented as a static scaffold of levers and pulleys, but in reality, it is a highly dynamic biological engine governed strictly by the laws of thermodynamics. To understand how it generates force, we have to trace its architecture downward. Whole muscles are bundled into smaller fascicles, which are packed with individual rod-like muscle cells known as myocytes. Pushing past the cell membrane of the myocyte, we find dense parallel bundles of myofibrils, and spaced along these myofibrils is the sarcomere. The sarcomere is the fundamental microscopic contractile unit responsible for every striated muscle movement in the human body. To decode macroscopic human movement, we first have to deconstruct the microscopic machinery of this single sarcomere. Motor neurons communicate with muscles using action potentials, strictly binary, all-or-nothing electrical signals. Yet our mechanical output is infinitely variable. We can generate the delicate, sustained tension required to hold a scalpel, or the explosive force necessary to deadlift hundreds of pounds. This variable force does not come from a variable signal. It is the precise biological consequence of frequency summation and the strict thermodynamic limits of the sarcomere itself. We will trace the chemical trigger of this contraction, correct a widespread misconception about how cellular energy drives it, and use electromyography data to prove exactly where these mechanical limits lie. Analyzing these specific chemical and mechanical transitions builds the foundational reasoning required for high-stakes clinical study. Contraction begins at the neuromuscular junction. The motor neuron releases the neurotransmitter acetylcholine, which crosses the synapse and triggers a rapid depolarization of the muscle cell membrane. This electrical wave dives immediately into the deep core of the muscle tissue, traveling through specialized channels known as T-tubules. The electrical wave hits the sarcoplasmic reticulum, prompting it to dump stored calcium into the intracellular space. Look closely at the actin filament. In its resting state, binding sites are blocked by a ribbon-like protein called troponomycin, pinned in place by troponin clusters. The newly released calcium ions bind directly to the regulatory subunits of the troponin clusters. That binding event causes an immediate conformational shift. The activated troponin physically twists the troponomycin ribbon, sliding it away and unmasking the actin binding sites. This unmasking process stays active only as long as intracellular calcium concentrations remain high enough to keep the troponin saturated. Calcium translates a fleeting electrical signal from the nervous system into the sustained chemical availability required for physical contraction. Now we shift focus to the thick myosin filament, sitting adjacent to the newly exposed actin sites. There is a conceptual pitfall regarding what physically powers the ensuing contraction. It is commonly assumed that ATP is burned to execute the power stroke. That is incorrect. This resting myosin head is pre-cocked, carrying ADP and an inorganic phosphate. It binds to the actin site, forming a crossbridge. The power stroke is triggered by the rapid dissociation of ADP and phosphate, forcefully bending the head to slide the actin filament forward. Following the power stroke, the spent myosin head is permanently locked to actin. A fresh ATP molecule binds strictly to force its detachment. This ATP is then hydrolyzed back into ADP and phosphate, recocking the myosin head for the next cycle. This specific detachment mechanism is why rigor mortis occurs. After death, ATP production ceases. Without fresh ATP to force detachment, the myosin heads remain permanently locked to actin, stiffening the muscle. Muscle contraction is driven by the physical release of waste byproducts. It is muscle relaxation that requires the continuous input of fresh cellular energy. Zooming out from the molecular scale, a single crossbridge cycle generates a microscopic amount of movement, but thousands of these crossbridges firing out of sync create macroscopic force over time. We define a simple twitch as the isolated mechanical tension resulting from a single action potential and its subsequent pulse of calcium. This graph maps tension over time. If a second stimulus arrives before calcium is cleared, tension from multiple twitches adds up. This frequency summation causes the tension to stair-step upwards until it hits a flat maximal force plateau known as tetanus. The speed at which a muscle can reach this maximal tetanic tension is strictly bounded by metabolic and thermodynamic constraints. This electromyography data plots the rate of tension generation against muscle temperature. At physiological temperatures between 25 and 35 degrees Celsius, the slope is exceptionally steep and efficient. As temperature drops below 15 degrees, the curve flattens and the tension generation rate plummets. This macroscopic data perfectly maps to the microscopic chemistry. Colder temperatures thermodynamically slow down the enzymatic hydrolysis of ATP inside every single myosin head. The cold also increases the physical time required for the ADP and phosphate byproducts to dissociate from the myosin head during the power stroke. This microscopic molecular bottleneck directly dictates the macroscopic lag in tension generation documented on the electromyography graph. Biological force is never unlimited. It is strictly bounded by the thermodynamic efficiency of the chemical reactions powering it. For that titanic force to do actual work in the real world, the muscle must pull against a rigid anchor. This brings us to the skeletal system. We often assume bone is static, dead scaffolding. A macroscopic look at a long bone refutes that immediately. The structure transitions from a dense, protective outer layer of compact bone into a spongy, highly vascularized inner core. At the microscopic level, the bone matrix is organized into Haversian systems, complex structural rings that deliver nutrients directly to living cells embedded within the bone. These cells carry out constant bone remodeling. This continuous turnover is driven directly by the physical stress and microfractures caused by repetitive muscle contraction. Here, multinucleated osteoclasts dissolve the damaged bone matrix. Closely following them are osteoblasts, which secrete fresh, reinforced bone material into the cleared space. Bone architecture exists in a constant, lifelong state of controlled destruction and recreation. It adapts its own density specifically to withstand the variable forces generated by the muscular engine. While essential for structural maintenance, the destruction of bone matrix by osteoclasts also drives the body's systemic chemical regulation. The skeletal system is the body's primary calcium reservoir. It stores the exact ions required to unlock the troponin complexes in the muscle sarcomeres. This supply is governed by an endocrine feedback loop. When systemic calcium levels fall too low for muscle function, parathyroid hormone triggers osteoclasts to dissolve bone and release stored calcium into the bloodstream. Conversely, when calcium is abundant, the hormone calcitonin stimulates osteoblasts to take the excess blood calcium and store it back into the hard bone matrix. Binary electrical signals from the nervous system command precise, thermodynamically constrained chemical reactions in the sarcomere. The musculoskeletal system operates as a closed biomechanical loop. ATP thermodynamics dictate the limits of muscular force, and that continuous mechanical force sculpts the living skeletal engine to sustain it.